All right, so we are recording. Um, we've learned a lot about bone tissue already. The structure of a long bone and a flat bone, all the living cells in bone, a little bit about how bone matrix is created and what the constituencies are. We talked about spongy bone versus compact bone and the differences, a few similarities. I'm going to start off with sharing a screen talking about the chemical composition of bone. So this is all about bone. I'm just writing chemical composition. In order to get through as much material and explanations as possible, I'm not going to write out every single word. I'll write headers and terms and then describe them to you and explain them to you. So there's basically two categories of the chemical composition in bone, and that is there's an organic component and an inorganic component. Strictly speaking, organic just means any molecule that contains carbon in it. But the organic compounds in bone are basically the living cells. That's part of the organic portion. And the stuff that is secreted by those osteoblasts. So I'm going to write plus osteoid. That is a comprehensive name for what is secreted. What's in this, to reiterate, collagen fibers are in there, glycoproteins, that just means a sugary protein. So that's the organic stuff. The inorganic parts are mostly minerals. Your book probably says mineral salts because that's just the form that those minerals are in, specifically calcium phosphates. That's the big one. So calcium and phosphate are the two highest in quantity of the mineral salts. But there's some other things as well. There's things like magnesium, manganese. This gives bone some flexibility. Okay? This really gives bone its hardness. So I'm going to just briefly talk about flexibility, the organic compounds, things like specifically collagen fibers. The collagen fibers that are found in this stuff that the osteoblasts secrete, they create bonds within the chemical. And when a bone is stressed, for example, if you get banged really hard or something gets bent, sacrificial bonds inside of osteoid, these collagen bonds, I'm going to write sacrificial bonds, often break at a microscopic level way prior to the bone actually breaking itself. So when we get banged really hard or our bone is subject to trauma, a lot of trauma, there are sacrificial bonds inside of the organic part, specifically collagen fibers, the organic part of bone. These little microscopic tears, if you want to think of it that way, occur. We don't even really know it. We may get a bruise, it may be sore, or maybe not. Obviously, if the bone is subject to enough trauma, the actual bone tissue itself can fracture or break. New topic. Before we go any further, we need to distinguish the difference between these two words, ossification and calcification. They sound the same. They're not really the same process. This refers to the laying down of calcium within a specific area. So calcium travels through the body. It's stored in bones, but it travels through the body and can be used by tissues when it's in the blood. So when calcium is traveling around in the blood, this word calcification refers to calcium being deposited into a specific area, okay, of the body, into tissues. That's calcification. ossification means the formation of true bone tissue. Now, in order for this one to occur, we need this process to happen first. So really, this is a required ingredient for this to occur. So we have to have calcium deposited into specific tissues or an area before ossification, true bone formation, can occur. Remember that story I gave you of here's an osteoblast, it secretes osteoid, which is this, the organic stuff is secreted by this osteoblast. So there it is, it's this sort of like gooey stuff. And then a blood vessel brings calcium to the area, and calcification is an ingredient. There's the calcium. And when these two mix, that's what hardens the bone and actually forms hard bone tissue. So when bone develops, when embryologically, we typically call that osteogenesis. Oh, let me go back to my black pen. So I'm going to write osteogenesis. A specific type of osteogenesis, embryonic bone formation. This is a long, drawn-out topic in your book, so I'll try to distill it down to the important qualities. Think of this. When we are developing in utero from embryos into a fetus, we start to grow skeletal structures prior to birth. That's what this topic is about. And there's two major forms of embryonic ossification or osteogenesis. All these words sound the same and they almost mean the same thing. So we're talking about forming bone tissue embryologically. And the two types are endochondral ossification. I don't have time to go over all of the nitty-gritty details, but I can certainly bring you up to speed on what this means. Let's just define this word. Chondral means cartilage. Endo means within. This means to form bone tissue. So the definition of this word literally means the formation of bone within cartilage, or in this case, within a cartilage model. So bone develops by replacing cartilage prior to birth. All bones use this type of ossification, of embryonic ossification. So I'm just going to write all bones except cranial bones and the clavicle, which is the collarbone, all right? So these are exceptions. And here's basically what happens. I'm going to draw pictures and just give you a step-by-step. If we start, what happens is we start in utero with a cartilage model. And so I'm going to draw this sort of cartoon-looking bone. That is kind of what it looks like. If you did an ultrasound and looked at a developing fetus, you'd see these skeletal structures, but they're not yet bone tissue. They're hyaline cartilage. So not fibrocartilage, not elastic cartilage, but hyaline cartilage is the model that's used to form all of our bones except these two. These are exceptions. So first what happens is a bone collar forms around the diaphysis of this little mini cartoon cartilage bone. So I'm going to draw that. That just means bone tissue starts to form here and here. In this area around the diaphysis, that's the shaft. Blood vessels invade this area. This is all happening in utero before we're born. So blood vessels invade or infiltrate the perichondrium. That just means around the cartilage. And they convert it to a periosteum, that tough fibrous connective tissue with real bone. And then mesenchymal cells differentiate into osteoblasts. Mesenchyme is an embryonic tissue that all connective tissues form from. I'm just going to write mesenchymal cells differentiate into osteoblasts. So really what we need to form new bone tissue is a blood supply, a rich blood supply. We need calcium, and blood actually brings the calcium, and we need osteoblasts to secrete osteoid. That's kind of the recipe for new bone tissue. These two bring, or the blood brings the calcium to the area, so we have calcification, and these secrete this stuff, and when these two combine, we get new bone tissue. Okay? So next, what happens after a bone collar forming in this area from blood and calcium arriving and mesenchymal cells giving us these things, the cartilage in the center of the diaphysis, so that the blue thing is all cartilage, hyaline cartilage, and the cartilage inside of the diaphysis starts to calcify. So the blood, what that means is the blood vessels actually penetrate, they take calcium in there, so it the material calcifies, and then chondrocytes, those are just cartilage cells in the shaft. They enlarge and sort of blow up and die, and they leave this space, this cavity in here. So cartilage cells blow up and die, we have blood infiltrating, and there are large spaces left by those dead cartilage cells. The matrix of the cartilage begins to calcify because of the blood's bringing in calcium. Then, step 3 is a periosteal bud invades internal cavities and spongy bone forms. So osteoblasts, mesenchyme differentiates into osteoblasts here. We have blood, we have calcium, now we have bone forming cells inside of there, and spongy bone gets laid down. By the way, this is worth noting, just for this entire chapter, spongy bone always forms before or prior to compact bone. Okay, that's worth remembering. So we get spongy bone forming in here. Originally, we have some spongy bone forming out here. Then this diaphysis elongates. The bone gets longer and longer. The medullary cavity forms from this hollow cavity. So this is the beginning of a medullary cavity in the center of the bone. Osteoclasts dissolve some of the spongy bone. Those bone dissolving cells start to dissolve some of the spongy bone in the center, and the medullary cavity actually starts to develop. Secondary ossification zones then appear in the epiphyses up here. So I'm going to draw blood vessels invading here and down here. So in endochondral ossification, first bone appears in the diaphysis, which is this area, the shaft, and then it starts to develop in the epiphyses here on the ends, here and here. The epiphyses then begin to ossify because same thing, we have blood vessels bringing in calcium. We have mesenchymal cells differentiating into osteoblasts. They secrete osteoid. Calcium is released from the blood, and like magic, we get new bone forming. First spongy bone, here and here. Then compact bone begins to remodel some of the spongy bone areas. So I'll draw that in black. All of this eventually becomes hard, compact bone. but the ends remain mostly spongy bone, except for a small thin covering of compact bone around here. So we end up with what is a fully formed bone with a medullary cavity in the center. Spongy bone remains in the epiphyses on the inside here and here. Compact bone remodels this whole diaphysis area. And last but not least, hyaline cartilage is left coating the surfaces on the ends so that we have that articular cartilage for a joint. So that's endochondral ossification. There's a second type, so I drew a number 1 here, big number 1. There's a second type that the cranial bones, the bones of the skull, and the clavicle use a different method of ossification in utero. So let me do that next. So I'm going to write it number 2, circle it. The second type is called intramembranous ossification. Remember, we're still talking about embryonic ossification. Remember, this is bones of the skull and the clavicle. What this term defines as is intra means inside, membranous means membranes. So inside fibrous membranes, we get bone forming. So that's its translation. It's a little bit different. We do not start with a hyaline cartilage model. We start with fibrous connective tissue. So fibrous connective tissue is just, think of something that looks kind of like areolar connective tissue. It's a bunch of fibers with some living cells. These are actually mesenchymal cells because the baby hasn't been born yet. So these are still mesenchymal cells in fibrous connective tissue. So I'm just going to write fibrous CT. And these are the steps. Ossification centers appear inside of this fibrous. Think of it like a bird's nest. That's what it looks like. And ossification centers appear, and these mesenchymal cells that I've drawn, they cluster together and differentiate into osteoblasts. There's chemical signals in the development of the fetus that convert these mesenchymal cells to, I'll use a different color for osteoblasts. So these differentiate into osteoblasts. So that's an ingredient that we need to form new bone. This forms what we call an ossification center. Osteoid gets secreted by those osteoblasts, that organic bone matrix, the part of the matrix, and blood vessels invade the area, bringing in calcium. So blood vessels invade this area. The tissue calcifies because calcium is released, and we ultimately get then ossification of bone, real hard bone matrix. A periosteum forms, that's that fibrous connective tissue, starts to form around the outside. What is, it looks like trabeculi of spongy bone forming in here. It's called woven bone at first. So I'll write that term down. If you read this, woven bone, it's just the first form of bone that looks like spongy bone that starts to get laid down in these ossification centers. A network of trabeculi then start to form around the blood vessels. So we get real bone spicules forming around these blood vessels. That's how trabeculi first form. Lamellar bone, that just means hard compact bone, then replaces the woven bone just deep to the periosteum. So we get this sort of hard lamellar bone encasing this network of trabeculi. What we end up with is that if you cut a flat bone, you have a layer of compact bone on the outside. This is the ultimate result. And spongy bone in here. And these blood vessels here remain interspersed in the spongy bone area. Red marrow ultimately appears in that cavity. All right, so those are the two embryonic forms of ossification. I need to now touch on bone growth. Oh, let me go to my black pen. First, we're going to talk about long bones in length, how they grow, how they get longer. So we've all heard of growth plates in young children, and that's what this is going to address. So if I draw a bone over here, here's a long bone. In a child, before in children and in adolescents, there is a cartilage plate that exists here and here in the metaphysis area. And technically, there's very little physical connection here and here to the bone parts. What I'm going to do is I'm going to magnify or enlarge this so that we can really look at the activity of a growth plate. Remember, a growth plate, you and I called a growth plate. The scientific term is epiphyseal plate. Okay, that just means growth plate. So at the epiphyseal plate, there's different areas of growth. So let me magnify, and what I'll do is I'm going to draw the top of this bone really big. This. Then I'm going to draw the epiphyseal plate here. And then I'm going to draw the diaphysis here. So we've magnified this area of our bone. The blue part is the epiphyseal plate. That is made of hyaline cartilage. So in young children and adolescents that still have physical growth to do, there are these cartilage plates at the ends of most of our long bones. And there are zones within that plate. And then there's another zone sort of here. And we're going to just label these 1, 2, 3, 4. That's actually in the growth plate or the epiphyseal plate. And the fifth zone that we're going to define is at the top of the diaphysis. So this is bone, this is bone, this is hyaline cartilage. And here's how a growth plate works. Zone number 1 is called the quiet zone. And you need to know what goes on in the quiet zone. The reason it's called the quiet zone is there's very little cartilage activity in there at all. So there's no chondroblast activity. Chondroblast means young cartilage cells that make new cartilage. So that's why this is called the quiet zone, because there's very little new cartilage growth, very small chondroblast activity. The second zone, number 2, is called, it's sort of the opposite, it's called the proliferation zone. This is where we have a lot of new cartilage growth. So we have a high chondroblast activity in the proliferation zone. So new cartilage is being, there's a lot of chondroblasts in here secreting new cartilage matrix. That pushes up this way onto the quiet zone. So the cartilage plate itself is growing this way, and it's pushing up on this diaphysis cap of bone. So we get cartilage plate growing in this direction, and the bone cap, the epiphysis, just keeps getting pushed up further and further, like this. That's because of this and this. Number 3 is called the hypertrophic zone. Hyper means over or more than. Trophic means to die or decay. So old cartilage cells in this area down here, they blow up and die. So these are older cartilage cells. They expand and they die and leave spaces in this older area of the growth plate. Number 4 is called the calcification zone. We already know what calcification means. I should write the word zone. In the calcification zone, blood vessels invade the area. There's still old cartilage down here, but blood vessels invade the area, and they lay down calcium from the blood vessels. So I'm just going to draw an arrow. Calcium gets put down. In layer 5, which is actually in the diaphysis, it's not technically part of the growth plate. That's called the ossification zone. In the ossification zone, we have a real high osteoblast activity. This one is chondroblast activity because it's cartilage. This one is osteoblast activity because it's bone. And these osteoblasts secrete their osteoid, their organic matrix, that combines with the calcium in this area, and we get bone actually growing this way as well. So as cartilage grows this way and pushes up on the quiet zone and that epiphyseal cap of bone, we get sort of new bone tissue proceeding behind it in this direction as well, where the old cartilage once existed. So this cap just gets pushed away, the plate keeps growing in this direction, and new bone forms behind it like this. That's how our bones grow in length when we're growing. At the end of puberty, so we're growing since we're born, we're going through childhood, adolescence comes, we get our growth spurt during puberty, which just means really high rate of growth activity. And I'll talk about that later. But at the end of puberty, hormonal signals tell this area, the proliferation zone in the growth plate, in the epiphyseal plate, to stop. So we get, at the end of our growth spurt, whenever it is, late teens or whatever, hormones tell the proliferation zone to stop making new cartilage. But this area, the ossification zone, keeps chugging right along. And so it eventually, the ossification zone eventually seals up bone on bone at the end of our growth spurt. And we get this remnant, this scar called the epiphyseal line. But that's really where bone fuses to bone. And the cartilage plate then in adults no longer exists. Okay, so that's how a bone grows in length. Now we're going to study how a bone grows in width. And that is termed appositional growth. I'm going to put in parentheses, width. This one's not quite as complicated. Osteoblast, so we have a bone again. I'm just going to draw the diaphysis of a bone. not the ends. Here's the medullary cavity. We have endosteum, endosteal tissue lining here. And we have periosteum lining the outside. It's thicker. Those are connective tissues. Now here's how appositional growth goes. Osteoblasts beneath the periosteum secrete bone matrix on the external surface of the bone. So osteoblasts that are found here on the surface of the bone secrete osteoid, and it calcifies, and we get the bone getting thicker here on the outside of the bone. So at the same time, osteoclasts, I'll make them a different color, on this surface dissolve bone matrix, and the medullary cavity gets wider as well, like this. So as these two activities occur, osteoblasts out here, osteoclasts in here, the end result is we get a thicker bone. We get a bone that's larger in diameter. This is the medullary cavity lined with endosteum. This is the periosteum on the outside. So we get a much larger bone in diameter. And please note, the medullary cavity is bigger as well. The reason is because osteoblast activity on the outside of the bone outpaces or is faster than osteoclast activity in here. So even though the medullary cavity does get wider, the overall bone gets thicker here because osteoblast activity outpaces osteoclast activity. Now, once we're fully grown, once we have finished our growth spurt, our bones continue to remodel throughout life, pretty much. And this term, bone remodeling, just refers to the ongoing process of coordinated osteoblast and osteoclast activity, even after we are fully grown. So in normal young adults, total bone mass remains, young adults meaning after you're finished growing, but until you're in your, say, mid-40s, late 50s, osteoblast and osteoclast activity remain constant. That means we do constantly have old bone being dissolved and new bone being laid down throughout our lives, but at the same rate. So our bones really don't change in size, overall size, unless we put some sort of stress on them, like well-tuned athletes, weightlifters, stuff like that. They have different bone mass due to the activities, the stress that they put on them. During remodeling, we have osteoblast activity, making new bone all the time, and osteoclast activity, dissolving old bone. I'm just going to write dissolve. The constant interchange of these two things is what we refer to as remodeling. The overall bone doesn't really change in size. This term, bone resorption, refers to this osteoclast activity. That's the process of dissolving old bone tissue. How that works really quickly, if this is the edge of a bone, osteoclasts are these big, ruffled, multinucleate cells that exist close to the edges of our bones. These osteoclasts are multinucleate. That means they have more than one nucleus. They're very large. I'm going to draw a big one just so I can illustrate what's going on. These cells secrete acids. So I'm just going to draw secreting a proton, a hydrogen ion is just an acid. So this cell secretes acid onto the bone surface, dissolving that hard bone matrix. The minerals that are, and other things that get dissolved enter the cell. So I'm going to draw calcium going in. It gets packaged in here, and then it gets released on the other side into the, it's called interstitial fluid. Ultimately, that calcium ends up in our blood. So the whole activity of these osteoclasts is that they dissolve through digestive enzymes and acids. They dissolve this hard bone tissue, and the constituents from that dissolving process actually go through the cell and into our bloodstream. That's called bone resorption. Now, the control of this remodeling process is largely based on how much calcium we need in the blood. So I'm going to talk about that next. I'm just going to write calcium storage and remodeling control. Okay, first, ionic calcium is the most used mineral in the human body, period. So I'm just going to write calcium and give you a short list of some of the things calcium is used for in the body. Muscle contraction. Every time a muscle contracts when we move, calcium is used. That includes the heart. Every time your heart beats, calcium is being used. Next, I'm writing nerve impulse. Every time a nerve sends an impulse transmission from one cell to another, it uses calcium to do that. Blood coagulation needs calcium. So you're bleeding either internally or externally. In order to stop that bleeding, calcium is necessary. Cell division, the process of mitosis. Every time a cell divides, typically calcium is used in that process. And finally, our glands, when they secrete things, calcium is involved in that process as well. So you can see why it's so important. 99, I think it's actually over 99%. 99% of the body calcium, it's even higher than that. So I'm going to put greater than 99% of calcium is stored in our bones. There's very little in our bloodstream at any given time. I think it's something like 0.001%. So a tiny, tiny bit of calcium is actually circulating around in our blood. The issue is, though, that amount is strictly regulated. That means if this amount fluctuates either too high or too low, it causes things, feedback mechanisms to occur in the human body. So its concentration in the blood is strictly regulated. Last thing I'm going to say is this: how we get calcium, we don't make it. Our body, the human body does not manufacture calcium. We have to get it from our diet. We have to ingest it. And calcium is absorbed in the small intestine after we eat it into the blood. And that's controlled by the metabolism of vitamin D. We went over vitamin D in the skin. Sunlight hits the skin, it makes a precursor to vitamin D, and then the kidney converts that to calcitriol. Well, that calcitriol regulates this, the absorption of calcium in the small intestine. So just think vitamin D regulates calcium absorption in the small intestine, okay? Now we're going to look at the role of parathyroid hormone. I'm going to abbreviate that from now on, PTH. What the heck is that? Well, our thyroid gland is located in our neck. Embedded in the thyroid tissue itself are these small, so I'm just going to draw a thyroid gland. It wraps around our trachea and the neck. On the back side of the thyroid gland, so the back of our neck, we have these little P-shaped structures. They're called the parathyroid glands. Para just means around the thyroid. Technically, they're kind of inside of that tissue. It's these glands that secrete this hormone, okay? We're going to talk about why that hormone is secreted and what it does when it is secreted. When calcium levels in the blood drop or decrease, so when they go down, when that strictly regulated amount of calcium in the blood decreases, that causes parathyroid hormone to be released. So I'm going to put PTH is released from those glands, right? And then PTH, that hormone, increases blood calcium levels in 3 ways. Okay, I'm just going to list them. First, PTH stimulates or increases osteoclast activity. You're probably thinking, okay, so what? Well, remember what osteoclasts do. Here's the side of the bone. Here's the osteoclast. It dissolves bone and dumps calcium into the blood. So that's how it increases blood calcium levels. It stimulates osteoclast activity to dissolve bone. That's calcium that's stored in the blood then is released into the blood, into the bone. So increases osteoclast activity. That basically unlocks the stored calcium that we have in our bones and puts it into the blood. The second way is PTH increases calcium absorption in the small intestine. So it stimulates, it's a co-hormone that, like vitamin D, it enhances calcium absorption in the small intestine. So the calcium that we're ingesting, we actually get more of it into the bloodstream. So that also increases blood calcium. And third, this hormone that's released from the parathyroid glands, PTH, decreases, so it's opposite, calcium excretion in the kidneys. What that means is it tells our kidneys not to release calcium in our urine. It gets reabsorbed into our blood, so it holds on to calcium so that we don't excrete it in our urine. That also helps increase the blood levels of calcium. So these are the 3 ways PTH increases blood calcium levels. So bones basically just act as their storage sites for calcium, and the osteoclasts unlock that calcium. Osteoblasts put that calcium into the bone for storage. Osteoclasts unlock it and release that calcium into the blood when we need it. This hormone, called serotonin, this is a hormone that regulates sometimes our sleep cycles as well as our mood. When we eat, serotonin is secreted into the blood and then into our bones, where it interferes with osteoblast activity. So I'm just going to write serotonin, a hormone that sort of blocks or interferes with osteoblast activity. So it sort of keeps new bone from being formed in minor ways. It's not super dramatic, but it's worth mentioning. New topic. I'm just writing bone and stress. I mean physical stress, not emotional stress. When a bone gets stressed, what happens to it? There is a scientist whose last name was Wolff who came up with this theory, so we call it Wolff's Law. What Wolff's Law states is that bone grows or remodels specifically in response to demands placed on it. So wherever there's stress on a bone, it activates new bone growth. I don't know if you've ever noticed this. If you're right-handed or left-handed, very dramatically, you may have noticed that your right arm, wrist, hand, or whatever, if you're right-handed, is slightly larger than your left hand. An exaggeration of that would be a tennis player. Professional tennis player who's right-handed, their right arm is noticeably larger than their left arm. Part of that size difference is due to muscle, but also the bone is bigger in that one limb that's used more. And this is sort of the process. So I'm going to draw the head of the femur in the leg. So there it is. This is the ball and socket joint. So there's the hip, the ilium of the hip. Here's the head of the femur. Whenever we walk, tendons that attach our leg muscles to this bone and what you and I would call hip muscles as well. These tendons, these muscle attachment points to the bone in order to move it, they put stress on the bone surface. So every time a muscle pulls, it stresses the bone in that area. That's actually a good thing because what that does is it stimulates bone growth. If you've ever had a broken bone and you've had to immobilize that bone for weeks to months, you've experienced bone atrophy, which means the volume of bone, the mass of bone tissue decreases. The reason for that is because the lack of this stressed activity on the bone, we actually lose bone tissue if it's not stressed or pulled on. So this physical stimulation stimulates bone growth. And there's a theory that most scientists subscribe to as to how, so I'm just going to write, if we stress a bone with physical stress, here's sort of what happens. First, it increases the electrical activity of the minerals in that area. So here's a piece of bone. There's a muscle attached to it. When the muscle contracts, it pulls on that bone, and the electrical activity of the minerals in this area right here is increased. What that does is it attracts the increase in electrical activity of minerals, attracts osteoblasts to that area. So we get more bone forming cells in that area. Osteoblasts secrete osteoid. I'm I'm just going to write osteoblasts ultimately are responsible, in conjunction with blood and calcium. Osteoblasts are responsible for new bone. So a weightlifter, for example, that stresses his or her bones in specific areas over and over, day after day after day, will actually have thicker bone mass in certain areas than somebody who does not. Likewise, highly tuned and highly trained athletes, a runner, for example, a marathon runner or even a sprinter that puts stress on certain areas of certain bones, they will have larger prominences like this in certain areas of their bones. Now, the cessation of that, the stopping of that activity causes the bone to decrease in mass as well. So I tell students all the time, some stress on our bones is good. Obviously, too much stress isn't. We don't want to break our bones from too much trauma, but walking up the stairs is much better for your bones than taking the elevator. And now you know why. Before I quit this lecture, I'm just writing other dietary effects. Bone. I'm just going to give you a laundry list of things and how they affect our bones. We've already mentioned a lot of these already. Vitamin D3, that's manufactured in the skin from the precursor called cholecalciferol. That's what our skin makes. Then this goes to the kidney. The kidney converts it, cholecalciferol, to calcitriol. That's the active form of vitamin D. Calcitriol then regulates calcium absorption in the small intestine, along with parathyroid hormone. So sunlight on our skin is good for our bones because it enhances calcium absorption in the small intestine. Vitamin C, magic vitamin, the wonder vitamin, also has an effect on our bones. It's necessary for collagen synthesis. So it increases collagen synthesis. Now you know that collagen fibers are a major component in the organic part of bone matrix. It also, vitamin C, enhances osteoblast differentiation, helps form osteoblasts. That's good for our bones as well. So I don't know if you've ever heard of this disease called scurvy. It's from the lack of vitamin C. I'm just going to write decreased vitamin C. Somebody who lacks a normal amount, a required amount of vitamin C in their diet, because we don't make that vitamin either, it has to be ingested. So it's vitamin C deficiency. It causes a loss of bone mass. So this disease, and the reason it causes loss of bone mass is we don't make as many osteoblasts if we have a vitamin C deficiency or collagen fibers. Sex hormones. That just means the steroid hormones that are in greater quantities in male and females, testosterone and estrogens. I'm going to put steroid hormones. Just think testosterone and estrogens. Both male and females have both types of hormones, just in greater quantities, that's all. We call testosterone the male hormone just because males make more of it, and estrogen the female hormone for opposite reasons. What they do is they increase osteoblast activity significantly. That's why during sex hormones at puberty are the key reason for our growth spurt at the end of puberty. So we grow since the time that we're born, and it's really growth hormone that affects our growth when we are children. Then when we enter puberty, sex hormones get secreted, estrogens and testosterone, in great quantities really quickly. And it's these that are responsible for our actual growth spurt. Growth hormone is actually responsible for, I'm just going to put childhood growth. That steady growth from being an infant through adolescence. Vitamin A does have an effect on our bones as well. It increases osteoblast activity. You can see how significant these vitamins are in our diet to healthy bones. And vitamins, I'm just going to write them on this line, vitamins B12 and vitamins K, they increase protein synthesis in our bones. I'm going to call that good for this lecture. I may try to supplement with a third lecture either tomorrow or Saturday, because I know you have a quiz coming up. Let me cut my recording here.